Good morning. I hope you're all doing well. It would have been great to have seen you in person this year, but I'm glad we have found other ways to communicate and engage with each other. In my presentation, I'll provide a summary of the work we have been doing in the last year, specifically focusing on some of the services provided to our forestry stakeholders, namely diagnostics, extension, screening and biocontrol. But I'm also going to discuss some of the recent progress on research projects, the application of which feed directly into those technical services. My objective is to illustrate the importance of the feedback between the services you see on this screen and the core focus of our group, namely to produce high-quality research to inform knowledge-based responses to tree health threats. Time is limited, so I can't discuss all our projects. Rather, I will highlight some examples of how research is translated for tree health management. I'm not going to have time to acknowledge everyone during the presentation, so I want to here indicate that the work presented is part of a fantastic group, including the students and staff of the TPCP, but also our various forestry colleagues in South Africa. I'll start with providing a summary of submissions to the Diagnostic Clinic. One submission, for example from a company or institute, can contain multiple samples from different sites and requiring separate diagnosis. As you can see from the graph, there were submissions of samples to the clinic throughout the year, with the obvious exception of April this year due to the lockdown and thus closure of the Diagnostic Clinic, and also less submissions in December due to the decreased field activities in the December break and the clinic closing for 2 weeks in that period. Interestingly, there was a very high number of submissions in February. If we consider the period from January last year to March this year, there were 1,300 samples received. The majority of submissions were eucalypt samples, but with a large proportion of pine and wattle as well. Many of the samples were related to post-planting mortality, as well as insect pest and samples sent from nursery hedges or for nursery screening. In addition, a number of submissions were plates containing isolated fungal cultures for identification, such as the pitch canker fungus. Of course, when it comes to important pathogens, such as the pitch canker fungus, a quick and accurate diagnosis can make the difference between being able to respond in time or not. However, some of the traditional diagnostic tools are either not sufficiently accurate or not sufficiently rapid, and thus the continued development and implementation of improved diagnostic tools is something of great importance. The recent development of a LAMP PCR protocol to detect the pitch canker pathogen has been such a development. LAMP uses isothermal amplification that is carried out at a constant temperature. The PCR is very sensitive and does not need a PCR machine for the amplification, as would a traditional PCR. Thus, it allows for very specific and more rapid diagnosis that is particularly suited for field-based detection. The LAMP primers were developed for the pitch canker fungus through a collaboration between FABI and the University of Georgia. These primers are superior to those published previously as they do not cross-hybridize with other Fusarium species, and thus provide a precise and rapid diagnostic tool for the pitch canker fungus, one of the most serious pathogens threatening pine forestry in South Africa and worldwide. These primers will now be tested in the field in both South Africa and the USA. Importantly, the development of these primers is an excellent example of how basic research has translated into a much-needed application. The foundational research that led to the development of these primers actually started about 15 years ago, when the QTL linked to growth of 2 Fusarium species was identified during the PhD study of Leishman de Vos. A few years later, the Fusarium genome was published by FABI, the first fungal genome to be sequenced in Africa, making it possible to investigate the genes that underpin the QTL. It was identified that the 5 genes underpinning the QTL were unique to Fusarium cercinatum. The 5 unique genes provided a large piece of unique DNA sequence from which to choose the perfect primers for LAMP. Interestingly, this same diagnostic tool is currently being considered for COVID-19. Moving from diagnostics to extension and monitoring, in the last year there were a total of 73 field trips, including TPCP and CTHB activities, giving a total of over 1,000 person days in field. This was across nearly all provinces of the country and includes consultations, monitoring, establishing trials, collecting material for research projects and other activities. These field trips include visits to sites severely affected by pathogens or pests. Some of the usual culprits were seen, but also some new or rather nearly forgotten pests and pathogens as well. In some cases, severe cases of drought, such as what can be observed here in the Greytown area, resulted in stress-associated pests and diseases. One stress-associated insect that was particularly abundant last year was the Mediterranean pine beetle, Orthotomicus erosus. Although reported in South Africa in 1968, this insect is generally not considered a primary pest. However, large areas of drought-stressed trees in Eswatini last year facilitated high infestations of this beetle. And I am sure many of you know about the widespread infestations of the wattle semilooper last year. This is a native insect with sporadic infestations on wattle, where the larvae of this moth, as seen in the photo, feeds on the foliage. Outbreaks of this insect on wattle have been reported before, but not for some decades. And in some cases, infestations were severe. Although most of our attention with regards to insect pests of plantation trees has generally been on the alien invasive species, this outbreak clearly emphasized the importance of native insects. This is especially considering that the drivers behind these outbreaks are generally not well understood, and management options can be limited. For example, the use of classical biocontrol is not an option as it is with introduced pest species. More research on native insect pests is thus needed. Other pest disease issues last year included species that you are likely very familiar with, such as Phytophthora, Gonipterus species 2 and Leptocybe invasa, as seen in these photos. The annual Leptocybe national monitoring, which involves forest industry, the ICFR and the TPCP, has shown that levels of insect pests such as Leptocybe invasa change over space and time, likely influenced by climate, establishment or non-establishment of natural enemies, and also changes in the eucalypt varieties planted in an area. These changing population levels of insect pests, including the sporadic outbreaks of native pest species, demonstrates the importance of monitoring these species to be able to predict outbreaks and timelessly introduce management interventions. However, for many of our insect pests, we have no efficient monitoring tool in place. And one of the main monitoring tools that can be used for forest insects is lure-baited traps. These traps use some form of attractant, for example stressed tree volatiles or a sex or aggregation pheromone, to attract the target insect. This has been used successfully in South Africa for the Sirex wood wasp in collaboration with the USDA some years ago. But for many forest insect pests, lures, whether based on tree volatiles or pheromones, are not available, and thus research capacity in chemical ecology to isolate, characterize, and test these components is essential. In previous TPCP meetings, we have discussed the successful development of a pheromone-based trap for the eucalypt cosset moth. In this last year, the highlight has been work from Lucky Marie Skeeper's PhD to confirm and test pheromone traps for the pine emperor moth. This is a native insect with sporadic outbreaks where the larvae can cause severe defoliation of pine trees. In these photos, you see the adult moth, about the size of an adult hand, and Jeremy Allison here, very happy that the traps and lures have been effective to trap these moths. Before the field trials, there was much research back in the labs, where GC-EAD studies confirmed that females produce a two-component pheromone, eliciting a major and a small response from male antennae. The field study in 2020 tested the dose response to this pheromone, confirming its effectiveness to attract the adult moths. Ongoing work is to determine the active space of the trap, elucidate the minor pheromone constituent, and test whether addition of the minor pheromone increases trap capture. The application of this research is that a monitoring program can now be developed for this pest. In addition, further research can investigate potential applications such as mass trapping, as has been done for the cosset moth, and mating disruption. This research developed from the foundation of chemical ecology established some years ago by Bernard, pioneered by Mark Bauer, and now with an applied chemical ecology satellite lab at FABI, which is led by Jeremy Allison. Perhaps the most significant current tree health threat in South Africa is the Polyphagous shuttle borer. This tiny ambrosia beetle was introduced to South Africa with its fungus. In about 10 years, it had spread all over the country, affecting 150 tree species across agriculture, native and urban forests. Currently, no PSHB infestations have been observed in any commercial plantations of pine, eucalypts or wattle. PSHB infestations have, however, been observed on roadside wattles, blackwood and a single eucalyptus tree. These were all on individual trees exposed to stress factors, but highlighted the importance to screen for susceptibility of commercial forestry species to this devastating pest. Fortunately, the TPCP CTHB group has some decades of experience in screening susceptibility to fungal pathogens, and Wilhelm de Beer and his team have now completed these trials, where 2 Fusarium isolates and a graphium associated with the shuttle borer were inoculated into wattle and eucalypts, and lesion lengths compared to a control with no fungus over time. Fusarium inoculations caused some streaking in wattles and eucalypts, but lesion lengths were not significantly different to the control, and no disease symptoms developed. There is thus currently no evidence that commercially grown species are threatened by PSHB. We cannot, of course, exclude the possibility that some untested varieties of wattles or eucalypts might be susceptible to this disease, or that susceptibility may develop in time. Thus, continued monitoring is needed. But what is also needed is to develop means to understand and predict changing patterns of susceptibility with changing climate, and Almut will briefly discuss this exciting development in her presentation. Changing the focus now to biological control, and specifically the use of Deladenus sircidicola, the nematode, to control the Sirex wood wasp. This last year, we produced and sent about 450 million nematodes to the TPCP members for the inoculation of about 3,750 trees, and dissected over 3,000 wasps to provide data on the parasitism levels across the country. One of the exciting recent developments is the discovery of a new lineage of this parasitic nematode in Australia. This was work from the PhD of Fiawut Eshetu in collaboration and support from the National Sirex Coordinating Committee of Australia, including Helena Rong and Angus Carnegie. We are now testing this lineage in the field to investigate if it can obtain better results than lineage B that is currently used for inoculations. It is through many years of establishing international collaborations and research to understand the diversity of these organisms that we are able to pursue such opportunities to improve current management strategies. Another ongoing project linked to both biocontrol and monitoring is assessing the distribution of the gall wasp Leptocybe invasa, including the 2 lineages reported in South Africa, and to assess the distribution and prevalence of its parasitoids. This is work involving the ICFR industry and the TPCP. Through the Leptocybe national monitoring, we have been able to track changes in the distribution and prevalence of the parasitoids. Even within only 4 years of data, from 2016 to 2019, we can observe substantial changes in distribution and prevalence of these parasitoids. For example, the parasitoid Quadristichus mendeli, in red on the figure, was unintentionally introduced into South Africa, possibly with another introduction of the pest Leptocybe. It was first confirmed in the country in 2016, and although not reported in the plantation areas until 2018, it is now the dominant parasitoid in some regions. This information enables us to reassess management strategies, for example, potentially redistributing Ophthalmus masceli to areas where it is currently not present to boost parasitism levels in those areas. And importantly, through the development of restriction enzymes, we are now able to track the change in distribution of the 2 lineages of Leptocybe invasa. This slide shows that Leptocybe lineage A was already present throughout the country when the national monitoring started. However, Leptocybe lineage B, which was detected later, has in just a few years spread to all the main plantation forestry regions. as discussed at previous meetings, we're investigating possible differences in host preference and interaction with biocontrol agents between these two Leptocybe lineages. But continued monitoring is important to detect potential introductions of new lineages or even new species. The eucalyptus night beetle continues to be one of the most serious pests in eucalyptus plantations in South Africa, as well as other countries in the world. Improving biological control is one of the research areas that we have been focusing on, but for experimental research on existing and potential new biocontrol agents, a reliable rearing colony is needed. This has proved more difficult than previously thought, so in the last year and in this coming year, our efforts are focused on investigating different factors that influence the development of Gonipterus and its parasitoid, and using this information to improve our rearing colony. This includes testing different media for pupation, as you can see from the slide, investigating optimal temperature and humidity for oviposition by the egg parasitoid Anopheles nitans, and so on. One of the interesting projects that Michelle Schroeder has started is to develop a mark-recapture method for the parasitoid Anopheles nitans. Fluorescent paint powder was used as the pigment to mark the insect with. An indirect marking method was used by coating the parasitized egg capsules with the paint powder. The insect will then obtain the pigment when they emerge from the egg capsules. Due to the small size of the insects, their flight behavior can be influenced if they are coated by too much paint powder. Therefore, a carrier medium, in this case psyllium husk, was mixed with the paint powder to reduce the amount of pigment that the insects will be coated with upon emergence. A number of different treatments have been compared. Results are only tentative, but from the initial trials, the treatment without the carrier, treatment 3 on the slide, performed the best with regards to the percentage of Anopheles wasps that were marked and the longevity of those wasps. In all treatments, the parasitoids were observed cleaning themselves, but the majority of parasitoids that were marked upon emergence, the mark was still visible when the insect died. It is possible that the psyllium husk powder was treated with pesticides, as this was a store-bought food product, which may have contributed to mortality of the insect shortly after emergence. Thus, further work is needed to confirm these results, and in addition, these methods will be tested in a walk-in cage experiment, and yellow sticky traps will be tested to determine whether they are an effective method to trap the marked insects to be released. The development of a mark-recapture tool would enable us to investigate the dispersal of the wasp and provide data that may help us to understand the variation of parasitism over space and time. This tool would also be useful to track the dispersal of newly introduced populations of Anopheles nitans, work that we are planning for in the near future. In this presentation, I have provided a brief summary of some of our technical services to our stakeholders and highlighted some recent applications within these services that have derived from our research. In some cases, this has been a 15-year or longer journey. Some applications have been born from specific applied research objectives linked to those applications. Host specificity and efficacy studies of biocontrol releases is such an example, as is research on insect pheromones leading to lure-based traps, mass trapping tools, etc. In other cases, such as with the development of the LAMP PCR, basic science to unravel the complexity of an organism opened doors to other questions, which when pursued opened doors to further questions, which in turn led to potential applications that were not necessarily considered or even thought possible at the onset of the research. These three aspects, basic science, applied science, and application, are all interdependent, and investment in all three aspects is needed to produce knowledge-based solutions for the tree health challenges of today and of the future. Thank you very much for your time.